Hello and welcome to Allentown Central Catholic's Honors Biology 2 course with Mr. Rosenberger. Today we will be reviewing some details about plant growth. The difference between plants and animals leads to the difference between their growth in a fundamental way. Most animals are characterized by determinate growth, that is, they cease growing after reaching a certain size. Most plants, however, continue to grow for as long as they live, a condition called indeterminate growth. Indeterminate growth allows a plant to continuously increase its exposure to sunlight, air, and soil.
When compared to animals, the way indeterminate growth is defined may sound like plants are immortal. This, however, is not the case. Angiosperms have three categories of life cycles: annuals, biennials, and perennials. Annuals have the shortest lifespan. Over the course of only a single year or growing season, they sprout, mature, reproduce, and die. Some examples are grain, legumes, and some wildflowers. Biennials take 2 years to complete their life cycle. The second year is when they reproduce and make seeds. These include parsley, turnips, and carrots. Perennials live for many growing seasons. These include trees, shrubs, and some grasses.
In my section of 31.7, the main ideas are about meristems and primary growth. So plants continue to grow because they have perpetually dividing and unspecialized tissues, which are called meristems. And these meristems divide when conditions allow them to, and they produce new cells and elongate, and they become specialized. Not all of them become specialized though. Some remain in the meristem and continue to produce new cells, but others, they differentiate and are incorporated into tissues and organs of the growing plant. Now we have apical meristems, and these are meristems. They're at the tips and roots and the buds of the shoots of the plant. Cell division in the apical meristems produce the new cells, which enable plants to lengthen, and that's primary growth. So you have the apical meristems that produce new cells that allow the plant to grow, which is primary growth. So the tissues that are produced by this primary growth are called primary tissues. The primary growth allows roots to push through the soil and allows the shoots to grow upward, which increases the plant's exposure to light and CO2. So although the apical meristems lengthen both the roots and shoots, there are also important differences in the mechanisms of primary growth in each system.
This figure illustrates primary growth in a slice through a growing onion root. The root tip is covered by a thimble-like root cap that protects the delicate, actively dividing cells of the apical meristem. Growth in length occurs just behind the root tip, where the three zones of cells or successive stages of primary growth are located. Moving up from the root tip, they are called the zones of cell division, elongation, and differentiation. These three zones of cells overlap with no sharp boundaries between them. The zone of cell division includes the root apical meristem and the cells that derive from it. New root cells are produced in this region, including the cells of the root cap. In the zone of elongation, root cells grow longer, sometimes to more than 10 times their original length. It is the cell elongation in this zone that pushes the root tip deeper into the soil. The cells lengthen rather than expand equally in all directions because of the parallel arrangement of circular bands of cellulose fibers in their cell walls. The enlargement diagrams in the figure shown at the left of the root indicate how this works. The cells elongate by taking up water, and as they do, the cellulose fibers, shown in red, separate somewhat like an expanding accordion. The cells cannot expand greatly in width because the cellulose fibers do not easily stretch. The three tissue systems of the mature plant, dermal, ground, and vascular, complete their development in the zone of differentiation. Cells of the vascular cylinder differentiate into primary xylem and primary phloem. The differentiation of cells, the specialization of their structure and function, results from differential gene expression. Cells in the vascular cylinder, for instance, develop into primary xylem or phloem cells, based on certain genes are turned on and are expressed as specific proteins, whereas other genes in these cell walls are turned off.
Since we talked about how primary growth affects the plant, next we're going to address the changes that secondary growth causes on a plant. So secondary growth occurs primarily in woody plants such as trees, shrubs, and vines. Unlike primary growth, secondary growth causes a change in both height and diameter. This can be seen in a tree, for example. Each year it gains a ring, which is growth in the diameter. However, a tree does not just grow laterally into a fat stump, it also gains height. This happens because of certain dividing cells in the tissues called lateral meristems. These cells are arranged within two cylinders identified as the vascular cambium and the cork cambium that run along the length of the roots and stems.
The vascular cambium. What exactly is it? The vascular cambium is a thin cylinder of meristem cells that lies between the primary xylem and primary phloem. Essentially, it is the main growth tissue in the stems and roots of many plants. It is responsible for the production of secondary growth, which is adding layers of vascular tissue on either side of the vascular cambium. More precisely, it produces secondary xylem inwards toward the pith and secondary phloem outwards towards the bark. The diagram on the right shows primary growth versus secondary growth, and you can see that secondary growth mainly affects the thickness.
During secondary growth, secondary tissues are produced. The two types of secondary tissues are secondary xylem and secondary phloem. The secondary xylem and secondary phloem are given rise by the vascular cambium, which is responsible for secondary growth. The main difference between the two tissues is that the secondary xylem is added to the interior, whereas the secondary phloem is added to the exterior. If you look closely at this picture, you can see where the two tissues are placed in woody plants. Each year this process occurs, and within each year the secondary tissues produced are larger in circumference than the previous year. This allows the vascular cambium to thicken roots and stems of the plant. Figure 31.8a in the textbook demonstrates this process in a woody Eudicod stem. One is able to see the larger circumference of the secondary tissues each year. The stem is much larger in year 2 than the stem in year 1.
Secondary xylem makes up the wood of a tree, shrub, or vine. A woody stem gets thicker over the years as its vascular cambium produces layers of secondary xylem. The cells of the secondary xylem have thick walls rich in lignin, which gives wood its hardness and strength. Annual growth rings result from the layering of secondary xylem. The layers are visible as rings because of uneven activity of the vascular cambium throughout the year. Secondary growth is interrupted during winter each year when the vascular cambium becomes dormant. Secondary growth resumes in the spring, and a cylinder of early wood forms. Early wood is made up of the first new xylem cells to develop. They are usually larger in diameter and thinner walled than those produced in the summer. A boundary becomes visible between the large cells of early wood and the smaller cells produced at the end of the previous growing season, producing distinct rings in the cross sections of tree trunks and roots. A tree's age can be estimated by counting its annual rings. Varying thickness of the rings reflect climate conditions and the amount of seasonal growth in a given year. Growth ring patterns in older trees are one source of evidence for recent global climate change.
At the start of secondary growth, the epidermis is shed and replaced by the new outer layer, cork. Cork cells, having thick, waxy walls to protect stems from water loss, are made from cork cambium. Cork cambium is a meristem tissue formed from parenchyma cells in the cortex. While the stem and secondary xylem thicken, the cork and cork cambium are pushed out and fall, which we see as the peeling bark on trees. This bark is everything external to the vascular cambium. Bark is made up of three things: secondary phloem, cork cambium, and cork. Youngest secondary phloem carries out sugar transport. Older secondary phloem and cork cambium die. Tissues and cork cells protect the stem until they also join the bark. Cork cambium keeps regenerating with the aid of secondary phloem to produce continuous supply of cork.
So my part of 31.8 is mainly focusing on the figure in front of you. And so basically the log on the left is showing the results of several decades of secondary growth. And the bulk of a trunk like this is mainly dead tissue. The living tissues in it are the vascular cambium, the youngest secondary phloem, the cork cambium, and the cells in the wood rays, which you can see coming out from the center of the log in the right figure. And the wood rays consist of parenchyma cells that transport water and nutrients. They store organic nutrients and they aid in wound repair. You can also see the heartwood in this photo is in the center of the trunk, and it consists of older layers of secondary xylem, which these cells actually can no longer transport water because they're clogged with resins and other compounds that make the heartwood actually resistant to rotting. And because of this, because the heartwood doesn't conduct water, a tree can survive even if the center of its trunk is hollow. Then you can also see the sapwood, which is a lighter color, and that's actually the younger secondary xylem that does conduct the xylem fluid, also known as sap. And this basically, the rest of my part is about the useful products that are made from wood, like furniture, musical instruments, paper, insulation, a long list of chemicals, alcohols, artificial vanilla flavoring, and preservatives. And what makes wood so useful is the combination of strength, hardness, lightness, high insulating properties, the durability, how you can work with it. There's actually no good substitute for wood, and this produces like almost making it the skeleton of all the land animals, as the book would say. And the book also says that wood is actually an evolutionary adaptation that enables the tree to remain upright on land and keep growing year after year. And that's how it becomes so big. And it's all because of wood. Thanks, guys. Have a good one.